Okay, so we just took a look at how peptide hormones are made in the cell and how they're secreted from the cell. Now let's take a look at their effect on the target cells. So this is what's going to happen after the hormone has gone into the bloodstream and reached its target. Okay. Um, all right, so a few things about peptide hormones. Peptide hormones are hydrophilic, which means that they're going to be able to dissolve quite easily in the blood, which is basically an aqueous solution, okay? That also means that they're not going to be able to cross membranes very easily, right? Because membranes, as you might remember, have kind of a lipid center. So it's going to be hard for that hydrophilic peptide molecule to just go right through the hydrophobic environment of the cell membrane. Okay, so, and we'll be seeing that in the upcoming slides. Peptide hormones have a short half-life, usually a few minutes, due to breakdown by enzymes. So there are enzymes floating around in the blood looking for hormones to break down, okay? Now that seems a little like inefficient. Why make a hormone and secrete it in the bloodstream just to have it broken down in a few minutes? But that's how our bodies work, and it makes sense because whatever signal you want to send out, you want that to be a short-lived signal so that it reflects the current state of affairs in your body, right? So for example, if your blood glucose level is low, you're going to secrete a hormone that tells your body, hey, blood glucose level is low, we need to bring that back up, right? But you don't want that that signal to last forever, right? Because then your body's just going to continue to work on bringing blood glucose back up to the point where it's going to go way up, way out of, way beyond its set point, way out of its acceptable range, right? So what you want to do is send out a brief signal, let your body respond to that, and then have that signal end so that you can then send out a new signal that reflects the new current state of affairs in your body, okay? Okay, so peptide hormones are going to bind membrane receptors, and that again is related to the fact that they're hydrophilic and that they can't cross the membrane, right? So they're going to need to bind to a membrane receptor in order to bring out responses in the target cell, okay? And then inside the target cell, once that peptide hormone has bound the receptor, it's going to lead to what we refer to as rapid cellular responses, so fast, kind of short-term changes in the target cell, okay? In other words, it's going to trigger what we call a signal transduction cascade. So this is what a signal transduction cascade is. So hopefully this is just a reminder. You should have learned about signal transduction cascades earlier in other bio classes. But basically the idea is you have this molecule that's floating around in this case, in the blood. It's going to come over to the extracellular fluid. How does it lead to a response within the cell, right? So remember that it can't cross the membrane because it's hydrophilic, okay? The way it does it is through signal transduction cascade, right? So you're going to have your hormone, which is indicated by this yellow diamond right here. It's going to bind to a membrane receptor, okay? And then that receptor is then going to activate molecules inside the cell. And those molecules are then going to interact with other molecules, and eventually that's going to lead to some kind of response within the cell, okay? Here's another example of or another diagram that sort of shows what a signal transduction cascade is, right? So it's called a cascade because it's a sort of a cascade of events, a series of events, right? So you're going to start with your signal. That signal is going to take an inactive version of some molecule and it's going to activate it, okay? That molecule is then going to work on an inactive version of some other molecule and it's going to activate that molecule and so on until eventually you reach some final actual action within the cell. So for example, changing a substrate to a product, right? So you're gonna activate some enzyme that is going to lead to some conversion of a molecule into another molecule, okay? I want to address / review some principles of signal transduction, okay? So I want to remind you that usually in a signal transduction cascade, the initial signal gets amplified, okay? So in this example, it's shown right here in this diagram. We have one ligand molecule, okay? Binding to one receptor molecule in the membrane, that's going to lead to the activation of some enzyme, okay? That enzyme is not just going to activate one molecule, it's going to activate multiple versions of the same, multiple copies of the same molecule, right? So it's going to activate in this diagram, these 3 balls right here, okay? Each of these 3 balls in turn is going to activate, in this case, 3 of these light blue balls here, okay? And then each of these light blue balls here is going to activate 2 of these dark blue balls here, right? So in total, we've activated, I don't know how many this is, like 18, I don't know. Dark blue balls in response to this just one single molecule of the ligand, one copy of that initial hormone, okay? And that's part of the reason that hormones can be so effective at such low concentrations, is that we have this amplification happening, okay? So the overall idea here is that you can have a small initial signal, which then leads to a large effect within the cell, because each step of that signal cascade, you're amplifying the signal, okay? I also just kind of want to review these 2 terms. So remember that earlier in a series of events, we refer to that as upstream. Later in a series of events, we refer to that as downstream. Okay, I also now want to talk about, okay, we've talked about how we get that signal across the cell membrane and into the cell and activating a bunch of molecules within the cell. But then what? What's the final result of that? Okay, so what I want you to do now is kind of see if you can rack your brain and see if you can remember what are some of the things that can actually physically happen inside a cell as a result of one of these signal transduction cascades. Okay, so go ahead and hit that pause button. Okay, so hopefully you were able to remember something, but here's a reminder of some of the kinds of responses that you can get inside of a cell as a result of a signal transduction cascade, okay? So one thing you could do is you could activate motor proteins, so you can, and that will then start moving things around in the cell, okay? You can change enzyme activity, either upregulate it, right? Make those enzymes more active, or you can downregulate enzyme activity, make those enzymes less active. That then is going to lead to more of certain molecules, more copies of certain molecules, or less copies of certain molecules, right? You're gonna, like, for example, speed up cellular metabolism or slow it down. You can activate membrane receptors and transporters, right? So that then is going to allow the cell to become more sensitive to other signals outside of the cell, or less sensitive. We're going to be able to either transport more ions or other molecules across the membrane, or fewer, and so on. And finally, you can lead to changes in gene activity and protein synthesis. Okay, so that's not one of the usual consequences of activating, of peptide hormones. Usually peptide hormones are going to activate these sort of faster responses.